Imagine falling into a rabbit hole, not into a wonderland, but into the mysterious and twisted world of Alzheimer's disease. A journey that begins in the quiet town of Marktbreit and spirals into a saga of discovery, controversy, and the haunting possibility of Alzheimer's disease transmission from person to person. Welcome to the Alzheimer Rabbit Hole. Buckle up as this tale takes you from the roots of medical history to the cutting edge of neuroscientific intrigue. Now, let's delve into how a mysterious disease named after a visionary doctor sparked a scientific journey with breakthroughs and debates. Alois Alzheimer, born on June 14, 1864, in Marktbreit by the Main River, was shaped by an innate curiosity despite the early loss of his mother. This curiosity led him to study medicine and psychiatry at the universities of Berlin, Freiburg, and Würzburg, igniting a passion for anatomy and the microscopic intricacies of the human body. His career took a defining turn at the Frankfurt Psychiatric Hospital under Emile Scioli, where he adopted advanced psychiatric methods. Here, he met Auguste Deter, a patient with symptoms of paranoia, memory loss, and cognitive decline that deeply intrigued him. Alzheimer's detailed documentation of her condition culminated in the discovery of plaques and neurofibrillary tangles at her autopsy, marking the first identification of what would later be named Alzheimer's disease. In spite of the revolutionary nature of his findings, the initial presentation was met with indifference. However, support from Emil Kraepelin, a renowned psychiatrist, propelled Alzheimer's research into the spotlight, enriching the field of neurodegenerative diseases. Alzheimer later moved to Munich, where he and his team engaged in extensive research on mental disorders. Personal tragedy struck with the death of his wife, making him a single father to their 3 children. In defiance of these challenges, his work continued to gain recognition, cementing the term Alzheimer's disease in medical terminology. Although dying prematurely in 1915, Alzheimer's legacy as a pioneer in neuropathology remains influential, continuing to drive advancements in neuroscience and deepen our understanding of psychiatric ailments. His life reflects a story of resilience, innovation, and an unwavering commitment to medical knowledge. Next, let's delve into the story of Auguste Deter, a name synonymous with the origins of Alzheimer's research. Auguste Deter was born in Kassel, Germany, on May 16, 1850. Despite growing up in a modest family and facing the early death of her father, she married Karl Deter. She became a housewife in Frankfurt, where they raised their daughter, Thekla. Described as content and harmonious, their life took a dark turn when Auguste started showing symptoms of dementia in the late 1890s. Her behavior included severe memory loss, paranoia, insomnia, and aggression, seeing threats where there were none. By 1901, her condition had deteriorated so much that Karl, overwhelmed by her care, admitted her to a mental institution in Frankfurt. There, Dr. Alois Alzheimer took a keen interest in her case due to her relatively young age and peculiar symptoms. Dr. Alzheimer meticulously documented Auguste's progressive mental decline, noting her inability to perform simple tasks or recognize her surroundings. She frequently exclaimed, "I have lost myself," illustrating her profound disorientation. She eventually became bedridden and died in 1906 from sepsis caused by an infected bed sore. A post-mortem examination of her brain revealed senile plaques and neurofibrillary tangles, confirming a diagnosis now known as Alzheimer's disease. thanks to advances in medical science. Years later, a genetic analysis of her case provided insights into the potential genetic aspects of her condition, although findings have been mixed. Auguste Deter's story remains central to Alzheimer's research, representing the profound human toll of neurodegenerative diseases and fueling the continued search for understanding and effective treatments. As we delve deeper into Alzheimer's research, we encounter tales of scientific intrigue and allegations of fraud, linking even to the chilling aspects of mad cow disease. The narrative darkens, with stories of scientists gambling on pharmaceutical stocks amid breakthroughs and breakdowns tied to the disputed amyloid hypothesis. The journey ahead promises more twists. Notwithstanding the significant contributions of Alzheimer's foundational work, our exploration through the history of research for Alzheimer's disease unveils complex layers of controversy. These include disputes over the validity of the amyloid hypothesis and the ethical concerns surrounding pharmaceutical investments. These challenges to established beliefs underscore the need for fresh perspectives in Alzheimer's research. We're now examining an innovative concept called the mitochondrial cascade hypothesis. Introduced over a decade ago, this theory shifts focus to the mitochondria, the cell's energy producers, suggesting that genetic and environmental factors shape mitochondrial functionality, influencing the onset and progression of Alzheimer's. This idea challenges the older amyloid cascade hypothesis, which posits that beta amyloid plaque buildup is the primary cause of Alzheimer's. In contrast, the mitochondrial hypothesis suggests mitochondrial dysfunction might initiate the disease, with amyloid accumulation being a secondary consequence. This new perspective is supported by modern diagnostic tools like brain scans and spinal fluid tests, which provide insights into the brain's changes well before Alzheimer's symptoms manifest. These findings help us understand how aging and mitochondrial function interplay with Alzheimer's development. The evolving definitions of Alzheimer's, recognizing stages like prodromal and preclinical Alzheimer's, align with the mitochondrial hypothesis, viewing amyloid buildup as part of a broader spectrum of brain aging, not its central cause. Against extensive research targeting amyloid, the anticipated breakthroughs have been elusive, prompting a re-evaluation of this approach. The mitochondrial cascade hypothesis offers a compelling alternative. By suggesting that enhancing mitochondrial function could be a proactive strategy, it opens up new avenues for Alzheimer's treatment, potentially allowing us to combat the disease before symptoms emerge. After exploring the mitochondrial details of Alzheimer's, we now shift our focus to the cholinergic hypothesis, which emphasizes the significant role of acetylcholine, a key neurotransmitter in memory and learning processes. The cholinergic hypothesis is central in explaining Alzheimer's progression, suggesting that the disease may be linked to the deterioration of cholinergic neurons and reduced acetylcholine levels. Think of acetylcholine as the brain's messenger chemical that helps with memory and learning, similar to how a phone transmits messages between friends. In Alzheimer's, it's as if the phone's battery is depleting faster than usual, leading to a breakdown in communication and, consequently, memory loss. This dysfunction may initiate further pathological changes, including abnormal protein activity and increased neural inflammation, complicating the disease's progression. Current treatments primarily provide symptomatic relief and do not stop the disease from advancing. Drugs such as donepezil, rivastigmine, and galantamine slow acetylcholine breakdown, slightly enhancing cognitive functions, but not tackling the root causes. Researchers continue to search for more effective treatments by enhancing cholinergic neurotransmission, safeguarding cholinergic neurons, and targeting the accumulation of amyloid beta and tau proteins associated with Alzheimer's pathology. Additionally, experimental methods look into regulating neurotrophic factors like NGF and BDNF. The cholinergic hypothesis thus sheds light on critical aspects of Alzheimer's, yet it also highlights the necessity for a multifaceted treatment approach due to the disease's complexity. Shifting from neurotransmitters to genes, we delve into Alzheimer's familial and sporadic types, piecing together the puzzle of its complex origins. Alzheimer's is a widespread brain disorder that makes it tough for many people to remember things, think clearly, and manage everyday activities. The disease mainly comes in two forms. Familial Alzheimer's is a rare type linked to specific genetic mutations like PSEN1, PSEN2, and APP. Sporadic Alzheimer's, the more common type, is influenced by a mix of aging, genetic factors, and environmental conditions. Researchers use advanced techniques like genome-wide association studies to dig deeper into the genetics behind Alzheimer's. These studies have unearthed a bunch of genes that could raise the risk of developing the disease, helping us figure out better ways to treat or prevent it. For instance, the APOE E4 gene is often found in the usual type that appears later in life, while genes like PSN1 and APP play a role in the less common familial form. There's also something called cognitive reserve. It's like your brain's ability to keep running smoothly even when Alzheimer's starts to cause changes. How strong this reserve is can depend on your genes and lifestyle factors, like how active you are or what you eat. Regardless of all these discoveries, developing effective treatments is still challenging. Some new drugs that target brain proteins involved in Alzheimer's have been approved, which is promising. Experts believe tackling Alzheimer's effectively requires a well-rounded approach. This includes understanding our genes, our environment, and how we live our lives. Staying physically active and eating well is crucial, especially for those at greater risk because of their genetic makeup. While we've learned a lot about the genetic links to Alzheimer's, which could lead to new treatments, there's still a lot more to uncover about this complex condition. We are about to unravel the mysteries of amyloid plaques and tau tangles, but our journey may take a dramatic turn as we explore the high-stakes world of pharmaceuticals. Next, we dive into the controversy surrounding a company accused of data manipulation. This situation sent shockwaves through the stock market and cast a shadow over Alzheimer's research. Stay tuned as we navigate through this entangled tale of science and scandal. Alzheimer's disease, the leading cause of dementia, primarily manifests through two biological markers in the brain: amyloid plaques and tau tangles. These misfolded proteins disrupt normal brain function and are critical targets in Alzheimer's research. Amyloid beta, or A-beta, derived from a larger essential protein, sometimes accumulates into sticky plaques that hinder cellular communication, contributing significantly to the disease. Genetic factors, head injuries, or conditions like diabetes can increase A beta production, leading to these damaging plaques. To counter this, treatments such as Aduhelm have been developed and approved, though their efficacy remains debated. Other therapies, like Leqembi and donanemab, show promise in clearing these plaques and slowing cognitive decline. Simultaneously, tau proteins, crucial for nutrient transport within neurons, can form tangles that disrupt internal cell transport, further impairing brain function. While no treatments directly target tau yet, ongoing research aims to stabilize cell structures and prevent tau aggregation, with emerging diagnostic tools enhancing early detection and treatment precision. Together, amyloid plaques and tau tangles impair neuronal communication, particularly affecting memory and personality areas. This leads to a progressive loss of brain function and the gradual onset of Alzheimer's symptoms. Thus, understanding these processes is essential for developing effective Alzheimer's treatments. We are stepping into a contentious chapter where the search for an Alzheimer's cure intersects with serious allegations of scientific fraud, unraveling a controversy that has shaken the core of Alzheimer's research and undermined confidence in cassava sciences. Cassava Sciences, Inc. has faced intense scrutiny, mainly from short sellers who are skeptical of the company's claims and early research data. Despite being in its formative years without established credibility, cassava saw its stock price surge in early 2021 after promising results from a trial of its Alzheimer's drug. The approval of a similar drug by Biogen further fueled optimism, catapulting cassava's stock fivefold from the beginning of the year to a peak of $135.30. However, the tide turned dramatically after Stat News published a critical analysis in July 2021 questioning the drug's efficacy. This scrutiny intensified when a law firm alleged data manipulation in a formal complaint to the FDA, leading to a steep decline in stock value and $100 million in profits for the skeptics. Currently, the stock stabilizes around $20. Central to the controversy is Huoyuan Wang, a key neuroscientist at Cassava, whose work is foundational to the development of their Alzheimer's drug, Simufilam. Serious allegations of falsifying data in 20 research papers essential to the drug's progress have emerged. An investigation by the City University of New York unearthed possible data tampering, casting doubt on the drug's clinical trials and questioning the integrity of the research driving Cassava's efforts to combat Alzheimer's. The plot thickened when Dr. Matthew Schrag, a neuroscientist, began investigating claims of foul play in the scientific narrative. His detective work initially targeted Cassava Sciences, but his investigation soon took a dramatic turn. Schrag's investigation quickly escalated as it led to a significant 2006 study by Dr. Sylvain Lesné, which had identified a specific form of amyloid beta, A-beta 56, as a key factor in Alzheimer's disease. This study has long steered the direction of research and funding toward targeting this protein. Schrag, however, discovered discrepancies in Lesné's work, including what appeared to be manipulated images. This revelation cast doubts on a substantial body of Alzheimer's research, potentially undermining the foundational amyloid hypothesis that has dominated the field for decades. These developments sent shockwaves through the scientific community, highlighting the complexities of Alzheimer's research and emphasizing the critical need for a broader investigative approach. It also underscored the importance of maintaining stringent ethical standards and rigorous peer review processes to uphold the integrity of scientific research. Adding to the controversy, new evidence suggests that amyloid beta might not be as central to the development of Alzheimer's as previously thought. Findings from brain scans and autopsies show that amyloid is present even in the brains of those without cognitive impairment, challenging its role in the disease. Moreover, drugs targeting amyloid beta have mainly been ineffective at slowing disease progression and sometimes cause harmful side effects. These insights advocate for expanding Alzheimer's research beyond the traditional amyloid-focused narrative to explore more effective treatments and support mechanisms for those impacted by the disease. Delving into Alzheimer's disease unveils the crucial role of the gut-brain axis, a vibrant communication channel between our digestive system and brain that might hold key insights into the disease. This discovery highlights the need to incorporate advancements in immunology to enhance our grasp of neurodegenerative diseases, spotlighting the adaptive functions of microglia in the central nervous system. Researchers confronting the challenges posed by aging and systemic inflammation on brain health have pinpointed 4 neuroinflammatory tactics that could shift the disease's trajectory. These methods include protecting the blood-lymphatic barrier, reducing inflammatory molecules, improving microglia neuron interactions, and managing aging cells' stress responses. The gut-brain axis emerges as particularly vital, with studies indicating that imbalances in gut microbiota could influence Alzheimer's progression. Experiments showing that fecal microbiota transfers from healthy mice to Alzheimer's models result in better behavior, and fewer disease markers suggest a significant role for gut bacteria in moderating neuroinflammation and cognitive functions. Additional research confirming different gut bacteria profiles between healthy individuals and those in the early stages of the disease points to the potential early identification of those at higher risk for dementia. This narrative continues to unfold, highlighting intricate interactions between our immune system, gut microorganisms, and brain health, paving the way for innovative research and potential interventions to slow Alzheimer's through insights into neuroinflammation and our internal ecosystems. Exploring the mysterious connection between the gut and the brain in Alzheimer's disease reveals a startling discovery that challenges our previous understanding. It might be possible for Alzheimer's to spread from one person to another. A groundbreaking study published in Nature Medicine explores this possibility. The researchers focused on individuals who developed Alzheimer's unusually early after receiving human growth hormone derived from the brains of deceased donors. This practice was discontinued in 1985 after being linked to Creutzfeldt-Jakob disease, a rare dementia akin to mad cow disease. The study found that 5 individuals developed Alzheimer's sooner than typical after this treatment. These findings hint at a potential link between the hormone treatment and the early onset of Alzheimer's, suggesting that the disease may be transmissible like other prion diseases. Despite these intriguing results, the likelihood of Alzheimer's spreading in this way is extremely low, and the disease cannot be contracted through casual contact. This research deepens our understanding of how Alzheimer's might evolve and the potential ways it could spread from one individual to another. Now, as we grapple with the haunting notion of Alzheimer's transmissibility, we're about to delve into the shadowy realm of prions, notorious for their lethal legacy and unyielding resilience. A 55-year-old woman in the Netherlands experienced rapidly worsening symptoms, such as memory loss, hallucinations, and speech problems, which initially seemed like Parkinson's disease. Sadly, she died within 27 months from a mysterious condition. Curious scientists preserved her brain using formaldehyde and set it in paraffin for further study. Years later, another group of scientists revisited her brain tissue, injecting it into mice. Remarkably, half of the mice developed symptoms similar to hers, showing that the disease agent had survived a rigorous preservation process meant to destroy most pathogens. Recent studies identified the cause as prions, notoriously robust proteins that misfold other normal proteins in the brain, leading to fatal diseases like mad cow disease and Creutzfeldt-Jakob disease. Prions are exceptionally resistant, surviving extreme conditions that would kill most germs and pathogens. For instance, they can endure temperatures up to 200 degrees Celsius or 392 degrees Fahrenheit and resist breakdown by enzymes that usually dismantle proteins. This resilience allows prions to stay infectious even after standard decontamination processes that should neutralize them. Consequently, prions on medical instruments present a unique challenge. They can remain infectious despite thorough cleaning, necessitating stringent decontamination practices in medical settings to prevent disease transmission. The potential for proteins similar to those in Alzheimer's and Parkinson's diseases to behave like prions complicates safety measures further. These proteins might adhere to surgical instruments and evade standard cleaning, underscoring the critical need for meticulous sterilization practices to combat the spread of these severe diseases. After we uncover the relentless nature of prions, let's pivot to another sinister force in neurodegeneration, the rogue proteins A-beta and tau in Alzheimer's. These proteins eerily mirror prion behavior in their relentless march through the brain. Alzheimer's has two main features: senile plaques and neurofibrillary tangles formed from abnormal A-beta and tau proteins. Like prions, these proteins can corrupt other normal proteins, setting off a cascade of damage. This process can propagate throughout the brain and may start with A-beta, subsequently affecting tau protein. Potential treatments could involve preventing these proteins from accumulating, disrupting the interaction between A-beta and tau, and removing the accumulated clusters. Initially, interventions might target A-beta alone, but addressing both proteins becomes essential as the disease progresses. Understanding Alzheimer's through the lens of prion-like behavior offers new strategies for combating the disease. For example, preventing the aggregation of these proteins or eliminating the harmful clusters could be key. Given their prion-like characteristics, medical practices must maintain rigorous sterilization standards to avoid inadvertently spreading these conditions. As our journey through Alzheimer's research concludes, we are reminded of our relentless pursuit of understanding and healing. Each development, whether a breakthrough or a setback, strengthens our resolve to decipher the complexities of the brain and offers hope in our ongoing battle against this and other formidable brain diseases.